Today we are going to discuss about Enzyme Database. Enzyme Database is a repository of information. Enzyme information consists of the Enzyme Database consists of repository of enzyme information. Consists of enzyme information, which is related to nomenclature of enzyme. It's a collection of enzyme functional and nomenclature data of enzyme. Clear? Collection of functional and nomenclature data of enzyme. Okay, clear. Enzyme database is primarily based on the recommendation of nomenclature committee. That is IUBMB, International Union of Biochemistry and Molecular Biology. Okay. It contains data for each type of enzyme. First of all, when we are talking about the enzyme database, these are the factors along with enzyme data is present. First of all, EC number, enzyme commission number. Second one, recommended name. Third one, alternative names. If any alternate name is present, we need to mention. Next, catalytic activity. Catalytic activity of particular enzymes. Next, cofactors. If any cofactors are available, then we need to, not only the person who is going to submit, they have to submit along with nomenclature information. Next is the sample enzyme entries. ID 1.14. Peptidoglycine monooxygenase. Say enzyme entry for say sample or example. Okay. Next. Here we are going to discuss about some of the major enzyme database. First of all, BOCD, Biosite Open Chemical Database. Biosite Open Chemical Database. It's a collection of, from the name itself you can say, it's a collection of chemical compound data. A collection of chemical compound data. Most of the compounds act as a substrates in enzyme catalytic activities. At the same time, some compounds serve as enzyme activators, some serve as a inhibitor, some act as cofactors. Likewise, also the chemical structures may be provided for majority of the compounds. It's not necessary, but same time most of the cases it was provided. The chemical structure may be provided. Then only the users can easily understand and assess the structure. Next, the next enzyme database is BRINDA or Brunswick Enzyme Database. It was reached through www.brinda.uni-colon.de. Through that we can reach the or locate this enzyme database. It's a comprehensive collection of enzyme. It's a comprehensive collection of enzyme and metabolic information. It was based on the primary literature. Also, this enzyme database contains data from at least around 83,000 different enzymes. Just 83,000 different enzymes from 9,800 different organisms. It was classified approximately 4,200 EC numbers. That means enzyme commission numbers. Okay. The data present in Brinda are extracted and evaluated from approximately 46,000 references. Next one, Enzyme Data Bank at EMBL. Say Expressive Enzyme Data Bank. Okay, the Enzyme Data Bank at EMBL contain 4,173 entries, and in 2003 it says Enzyme Nomenclature Database. Also, the current version currently contain current version of EMBL Enzyme Database contain 3,700 enzymes around. Okay, it gets reached through express.ch/enzyme. Next one, enzyme. Which means integrated enzyme, integrated relational enzyme database. Also, it's a version of enzyme nomenclature. The is supported by IUB-EMB and contains enzyme data, and the enzyme which was present in the integrated relational enzyme database was approved by IUB-EMB committee. It was reached through www.eba.ac.uk/integrated enzyme. Next, kinase pathway database. Here, the kinase pathway play a major role in the regulation of cellular functions. It contains various kinds of information about the molecules, which is used to understand the signaling pathway and characteristics of organisms. contains the classification of protein. This database contains the information about the classification of protein kinase and their functional conservation, protein-protein interaction, protein-gene interaction, protein and the compound interaction, domain information and structural information. The protein, gene and compound interactions are automatically extracted from abstracts by NLP, Natural Language Processing. It may reach, that means the kinase pathway database reached through kinase dp.ontology. It's a kinase pathway database. Next one, PASPA-based database. It's a protein phosphatase information resource. It's a very first public resource which is dedicated to the protein phosphatase. It is this enzyme performs dephosphorylation reactions. Also, these enzymes, phosphate-based enzymes are implicated in many human diseases like diabetes, cancer, neurogenetic disorders like that. The main aim of phosphate base is centralize the growing base of knowledge in phosphatase research domains. Next, PKR, protein kinase resource, which play a major role in all major cellular processes. For example, signal translation, cell differentiation, cell proliferation, and cell cycle progression. Next, precise. Next database is the precise, predicted and continuous interaction sites in enzymes. This database contains the information of interaction between the amino acids residue of an enzyme and its ligands. It can reach through precise .bu.edu. The current versions of precise contain all information on interaction is extracted from enzyme ligand complexes, which was present in PDP database, protein database. It was extracted by using 4 steps. First one, proteins have the same GC number and all the sequences are similar. Second one, selecting a representative chain for each cluster. Third, selecting ligand types. Fourth, finding non-bonded interactions and hydrogen bonds. Fifth one, summing a interaction for all chains within the clusters. Next, Scope EC, Scope Enzyme Commission. It's a protein catalytic domain database. There are two major problems needed to be overcome to create these databases for sequence EC numbers and protein data bank structures. Next one, TECR-DB data bank, thermodynamics of enzyme catalyzed reactions database. Thermodynamics of enzyme catalyzed reactions. Contain comprehensive collection of thermodynamic data. The results from 1000 published papers which contain 400 different enzyme catalyzed reactions. Next, finally, Ergo. Formerly, it was named as with WIT. It's providing information about the functional role of enzymes.